How frequent do you have this seminar?
Once a month.
Once a month, okay.
So monthly seminars, and we started in September, just after the virtual Cancer Cachexia conference. And the idea was to keep the attention high, and it seems it is working. Hi, Andrea, I notice he's also connected. We have, I would say, young, but not that young board of educational committee, trying to invite top scientists like you, and we had great talks so far. We have a program for the following talks. Maurilia indeed is going to give I think it's the next talk. Maurilia, can you confirm? Yeah, in February is going to talk Maurilia Silander. So in the meantime, I can also advertise them for the people that are already connected. Then we are going to have Chris Koss in March from the Ohio State University. And then Laura Bindals, I don't know, Marco, whether you know her. She's working on microbiota, and she joined the cachexia community in the last years. And then in May, we're going to move to a clinical side of cachexia with Carla Prado from Alberta, and maybe talking about the diagnosis and the importance of skeletal muscle mass in cachexia and hematotoxicity and so on. So we're trying to be comprehensive, instructive. For sure, I know you're a really good teacher, so we have also young people in the audience. For the educational purposes, I think you're gonna be right for the scope. Yeah, for sure, Marco. Okay, so a couple of minutes more. It's 1 minute after the official time we should start, and then we will start with the seminar. We reached the 59 attendees, so that's great. Andrea, do you remember the previous seminars?
Yeah, it was around 60 also.
Yeah, so that's great. 60 now. So the interest is nice. There are many people following. So we are building up a community that is following. And in this period in which in-person meetings are not possible, I think it's a good option to stay updated and stay connected. I noticed that there are also people that we generally do not see at the two main conferences on cachexia. So I think it's a great opportunity. Okay, so 70 people, great. So I think I can start introducing Marco. So in the meantime, maybe other people are connecting. So welcome to everyone to this Cancer Cachexia Society seminar series. Today we are with Marco Sandri from the University of Padova in Italy. A very brief introduction. I'm pretty sure that most of you know him and know the research that Marco performed in the last 20 years. But let me just very briefly highlight some milestone to know him, especially for the young, since for me, Marco was a person that inspired me. And for the career of the young, I think you gotta have some idol. Sorry for exaggerating, Marco, but you're really one of the idols in the muscle field more than in cachexia. I would say more related to muscle physiology and pathology. And Marco is an MD and then had an internship in pathology. So from the basic study of medicine and pathology, he had a postdoc where he really exploded. And because he was in one of the best lab he could choose. He was in Fred Goldberg's lab, and he published really astonishing data. Marco was the first to re-highlight, sorry for telling a re-highlight, because autophagy and the lysosomal degradation had a first era, but it was one of the first telling that they had the relevance in muscle physiology and in the control of muscle mass. And he also collaborated with Bruce Spiegelman. So top scientists to have a new top scientist. So Marco is the next generation beyond those great scientists. And then he came back to Italy with a Talenton fellowship, if I remember right, Marco. But then he rapidly increased his affiliation. So he's affiliated to the Dulbecco Teleton Institute, to the University of Padova, even the McGill University, if I'm right. And now at the Department of Biomedical Sciences, he's the director, he's the dean of the department, and he's keeping on being a great scientist. So thanks, Marco, for accepting our invitation. And I don't want to steal more time, and I leave the floor to you, and please.
Thank you, Fabio, for really kind words. I'm not an idol of anyone, but I like to stay with the guys and to talk about science. So for me, it's a pity that we cannot stay alive, be together, because I really like to talk with people. Only talking with people, you have new ideas or new views of science. And today I want to share with you unpublished data about how I see some of the topics that are important, or some of the pathways that can be relevant, not that are unique, but can be relevant for cachexia. Usually I would start with this picture, an electron microscopy picture of a cell. This is a liver cell, a beta cell. And usually electron microscopy is very useful because you can have an idea how is the structure of the cell, how dense of organelles, of proteins is in the cell. And as you can notice, in our liver, let's change a little bit the option. There are space between organelles, ribosome, mitochondria, in the plasma reticulum in the liver. But if you look at the muscle, there is no space between all the organelles and the different proteins. So there's a very dense packaging of structures in muscle fiber. This means if you extrapolate that when you lose muscle mass, you have to remove proteins and organelles. So the balancing between degradation and synthesis now is more towards the loss of the components. And this happens when it's too much. You have, and can be not counteracted by nutritional approaches or other kind of approaches. Now you have a condition that is named cachexia, and this is, of course, a bad prognostic index. It increases mortality, and in some other disease can also increase morbidity. So 30% of the cancer people die for cachexia, and I think that most of you are familiar with this data. But there's another aspect which is important, that muscle is not only muscle loss. The problem is that you have also weakness and fatigue. So people, you can be very thin people with small muscle, you can still have a pretty strength in your muscle, you can have a normal life, but when strength and fatigue happens, that is something that is bad, because your independency or your quality of life dramatically decrease. And to talk about fatigue, you have to think about some physiology. So everything started with the motor neurons that depolarize or release the mediators, acetylcholine, that induce the polarization of fiber. And the polarization now is moving towards the cytoplasmic reticulum and is inducing the release of calcium. And now the calcium induces the actin-myosin interaction. Then cancers to be removed, and then you can have a relaxation. So components affecting this condition which affect all these components, they can promote fatigue. And the other aspect is the metabolic aspect. So to induce contraction, you need energy. Energy are coming from different sources, free fatty acid, glucose, or other kind of amino acid, and also oxygen. And mitochondria are using this fuel to produce ATP. At the same time, there are some other metabolites produced from the different pathways, like pyruvate, like phosphate, that can impact, can affect also the property of myosin-actin interaction, changing the pH or affecting the ability of calcium uptake and release. And of course, if mitochondria are not working, they can also release other mediators like oxygen, reactive oxygen species, and can also release other mediators that can impact on myosin-ductin interaction. So if you have to say which are the major components in muscle fiber that can affect force generation and also fatigue, you can have an aspect that is motor neuron and muscle interaction, metabolized confounding ATP production from mitochondria, and all what are affecting the ions, including especially calcium in this case. These are three important systems or three important components that really affect muscle fatigue. And this story started from when we started to analyze the muscle biopsies from cancer people. So we are a good collaboration with our surgery department, and we can get a rectus abdominis biopsy from people that mainly have pancreatic and colorectal cancer. So now I show some data coming from this biopsy. So now at the moment we have more than 100 patients biopsies, 23 control, and other are cancer patients. And these patients can be in already cachectic or in a pre-cachectic condition according to QM5 definition. And when I was looking at the morphology of this section, you can appreciate that the cancer people, they do have a reduction in fiber size, but at the same time, there are some features that has a pathologies I can recognize that are not only atrophic fibers. So these are flattened, angular-shaped fibers are typical fiber that happens when you lose even motor neurons or innervation. So this was really a peculiarity that I did also observe in other animal models of cancer cachexia. And we were wondering whether if one of the important trophic factor, motor neurons and neuromuscular can be affected by cancer. So one of the ways which you want to look if there is any alteration in a muscular junction, you can look at the localization of this protein count that usually stay adjusted at the postsynaptic side in muscle fiber. But if you lose motor neurons, now these proteins start to relocalize on all the muscle fiber in the cytosol. And indeed, when we look at muscle biopsies about non-CAM positive fiber, we do appreciate these angulated fibers more around the fiber that start to be NCAM positive, suggesting further that these small fiber are denervated fibers. And this is a quantification. It already started in pre-cachectic cancer. So keep in mind, these are a pancreatic cancer patient in colon that are developing very, in a high percentage they develop cachexia. So then we look at other kind of markers of denervation, like the expression genes that are usually upregulated in muscle denervation, like myogenin or RANX1. And again, we do see that during cancer patients, we have an upregulation of denervation program. And then we start to look in the blood if there are other markers of neuromuscular junction remodeling, like a solid cleave agrin that usually is cleaved at the muscular junction and is released and got in the blood. And indeed, that was an increase again in the blood of agrin, or NCAM, the other soluble fragment of NCAM that is still increasing the patient. Altogether, this data at molecular, morphological, and biochemical suggest that neuromuscular junction is affected when cancer starts to grow in person. Of course, to understand the mechanism inside, we moved to animal model, and we started with a C26 cancer tumor-bearing mice as a model. For people that are not familiar, this when injected C26 colon cancer cell in an animal, let's say in 4 weeks, they start to lose the body weight, and mainly they lose both fat and also muscle mass. And this is typical what's happened to the animals when we get cachectic by the syphilis colon cancer growth. And again, we start to look at if in this animal model, we do see what we have seen in patients. So NCAM, again, you see these flattened and angulated NCAM positive fiber that happens that are increasing in tumor-bearing mice. And we look at different genes that are induced, like myogenin, RANX, that also mask and CAM, and these are all upregulated in C26 tumor-bearing mice. And we start to look at more details about the morphology of neuromuscular junction. So we start to stain the postsynaptic side by bungarotoxin in blue, and then the presynaptic and axon in red, synaptic vesicle 2 and neurofilament. So you can appreciate pretty well in a whole muscle that there is the axon, and they reach the presynaptic side in yellow, and then you have a pretzel-like shape of the presynaptic side in which acetylcholine receptor are present. But in C26, now you do see that now the postsynaptic side is present, but you lose the presynaptic and the axon. This is a morphological observation, but I like also to see some functional data. So we asked to our electromyograph, and this is a quantification is important. As you can see, you start to have the loose innervation and start to increase the innervation even before you can get the muscle loss. So these changes, again, seems to appear before cachexia onset. So in the meantime, the tumor is growing. So I want to see also some data about electrofunctional, say, and so we look by electromyography, how is the compound muscle action potential. So we stimulate the nerve and we record the depolarization events in muscle fiber. And it has some amplitude. And here you have different, so you can stimulate more times and you can record the different CMAP. And by time you stimulate it, you lose or you decrease the amplitude in wild-type animals. So yeah, that the neuromuscular, there is a fatigue in neuromuscular junction transmission of the electrical signal. But if you have a cancer, now look, you have a dramatic decrease in the amplitude of the CMAP, suggesting that you have a problem in neuromuscular junction transmission. The CMAP is completely abnormal in shape. And then you have a decay in the amplitude that is pretty dramatic compared to the wild type. So this you can quantify in the amplitude decay. So for starting from 100% in the wild type after different stimulation of the nerve, you have some decrease. But if you have a cancer, you have dramatic decrease in the amplitude. And this can contribute to, of course, the weakness and the fatigue and all that, because if the neuromuscular junction is not working, now you have a less depolarization, less calcium release, and then, of course, less force. And keep in mind that the calcium is important for the mitochondrial shape. So the question is now, the alteration of muscular junction we do see is just because there's a tumor in animals, or is it because they have a cachexia onset? So for this analysis, we use a colon cancer cell line that is able to grow, but does not induce cachexia in animals. So you can see that when you implant the MC38 cell line in animals, the animals and what they implanted one on the sham, they still maintain the body weight during time, even if the tumor is still growing. So there is no loss of muscle mass by the MC38 tumor growth. TBI inferior gastropnea, CDL solid, do not have a difference in the muscle weight. And there is no alteration in genes that are relevant for the innervation. There are markers for innervation. So even if there is a tumor that is up to growth, there is no alteration in muscle mass and the neuromuscular junction artificial innervation program. So we start to work out to think about, okay, so alteration of muscular junction can happen independent from tumor growth, but when we have a condition of cachexia. And maybe muscle is in some way important in muscle, in the muscular junction maintenance. And that came out also because when we block autophagy, specifically in muscle, we do see alteration in our muscular junction and denervation. So we start to think that maybe there are factors, cytokines, that from muscle or from other sources can impact on the muscular junction. And between the different cytokines, we focus on one that was also induced in our autophagy deficient mice, that is noggin. Why noggin or BMP pathway? Why this? Because this pathway we recently found together with Paul Grigorievich, that is important for controlling protein synthesis, but also protein breakdown. So this pathway is able to activate some transcription factors that are important to repress the induction of muscle-specific ubiquitin ligase, like ATG1 and MURF1, and also a novel ubiquitin ligase that is named MUSO1. And these are important for protein breakdown. At the same time, are able to activate genes that are involved in protein synthesis and probably in mTOR regulation. So if this path is blocked, of course, we have exactly what we expect, an increase in protein regulation and decrease in protein synthesis. So we'll look Just to show you that noggin per se, when we overexpress in muscle, in wild type muscle, is sufficient to induce muscle atrophy. But if you have already a catabolic condition, like a denervation, muscle almost disappears. You have an exaggeration of muscle loss if noggin is overexpressed, suggesting that per se noggin can trigger a cachectic phenotype in muscle. Is an exaggeration of muscle protein breakdown. So indeed, when we look at noggin expression, we identify that in a tumor-bearing mice, noggin was induced as transcript, but importantly, it has a protein as well. So when we do the ELISA kit and we measure noggin protein muscle extract in cachectic mice, there is a significant increase in protein level. And this leads, since noggin is an inhibitor of BMP pathway, leads to indeed a decrease in the phosphorylation of the downstream target, so transcription factors MAD158. It usually is activated. If you block the BMP pathway, they are less phosphorylated. And this is the case when you have cancer to C26 tumor-bearing mice. Now, phospho-MAD1 is significantly decreased. Importantly, in the patients that we have, we do see a significant, mild but significant increase of noggin expression in precachectic and cachectic patients. So the point is, I show you that noggin is induced. If you overexpress noggin, you can have atrophy. Is the neuromuscular junction affected by noggin of overexpression or induction? Again, we overexpress noggin muscle wild type animals are in control, and we inject just the AAV virus, and we stain for the axon, the presynaptic side, and postsynaptic side. And in normal animal, this is a whole mount staining of tibialis anterior, and you can see that there is a perfect match in the control animal. But when you overexpress noggin, again, you lose the presynaptic and the axon, suggesting that noggin for BMP inhibition is sufficient to induce denervation phenotype. And when we look at the compound muscle action potential, we do see again the abnormalities in CMAP, as we did see in our C26 tumor-bearing mice. But importantly, we do see also spontaneous depolarization events that is typical of the denervated muscle. And altogether, we say that now noggin is sufficient to induce atrophy and denervation in animals. So in some way, cancer growth in a mice is important, or probably in humans as well, is triggering a noggin upregulation that blocks the BMP pathway, leading to a double hit. A problem of protein breakdown, inhibition protein synthesis, but at the same time, alteration of muscular junction. And altogether, they can really impact on the quality of the muscle and fatigue. If it is true, by overexpressing and reactivating the BMP pathway, we should now prevent all the features I've seen, so muscle loss and alteration of muscular junction. So that was experiment. We overexpressed BMP7 in C26 tumor-bearing mice, and all constitutively active type 1 receptors, so that they are separate downstream the pathway, IL-3. And as you can see, if you have expressed the type 1 receptor or the BMP7, we completely prevent the muscle loss induced by cancer in tibialis, as well as in gastrocnemius in two different muscles. Importantly, and this is even more interesting, as I show you, C26 tumor-bearing mice, they do have a problem in the amplitude, in the shape of the MAC, in the amplitude, and in the decay of the MAC, or in the deposition events. But if you restore BMP7 or reactivate the pathway, now the amplitude is, the shape of the MAP is restored, the amplitude is restored, and the decay is significantly reduced. You maintain the action potential, the depolarization events in muscle fiber. So altogether, they say that this, and this is the quantification, by cancer, you do have a decay in the amplitude, and by BMP7 overexpression or the active IL-3 overexpression, we maintain the amplitude of the action potential in muscle fiber after nerve stimulation. So noggin seems to be, and BMP seems to be really impacting the neuromuscular junction and the neuron and the synapse, and so the electrical stimulation seems to be affected. So why noggin is inducing cancer detection? So we use again the two models, the colon, the mice that have a colon cancer line that do not develop cachexia, and the animals that do have colon cancer cell line, but they develop cachexia. And we look in the serum, two important layers that have been in the past shown to affect muscle loss. Activin A, actually there was no significant difference. Both have upregulation, or interleukin-6. And now we do see that only the C26, the animals that have C26, they have in the blood high level of interleukin-6. Not only in the blood, but even the muscle, they have an upregulation of interleukin-6 transport level. And that is an important aspect that I will come back to later. So we started thinking that maybe interleukin-6 signaling is what is important for noggin upregulation in C26 tumor brain mice. And indeed, when we express IL-6 in muscle of wild-type animals, but not when we express another pro-inflammatory cytokine like TNF-alpha, we do see that noggin was induced, suggesting that just not only inflammation, but specifically IL-6 and the pathways downstream IL-6 seems to be involved in noggin upregulation. And so IL-6 is able to bind to the receptor and activate a cascade of events that phosphorylate STAT transcription factors. So we start to think that maybe this pathway is important for noggin upregulation. If this is true, by blocking STAT, by knocking down STAT, we should revert or blunt noggin upregulation. And that was an experiment. When we express IL-6, we have a noggin upregulation. When we knock down by citrulline STAT3, now we blunted the upregulation of noggin induced by IL-6. Moreover, by chromatin precipitation, we identified that noggin promotes several STAT binding signs to putative, but especially in this region from 5 to 14, there's an increase, a significant increase in the binding, so in the recruiting of STAT on noggin, STAT3 on noggin promoter. Again, another evidence that noggin is under control of IL-6 STAT3 pathway. So to summarize this first part of my talk of this preliminary published data, is that the cancer, the interaction between cancer and the host difference, like, induce changes, different changes. It's not only one pathway, when we said at the end, it's not one pathway, it's many different signaling pathways are activated and that induce secretion of different cytokines. For instance, cancer can inject C26, induce activin A and interleukin-6. Interleukin-6 is able to activate in the receptor STAT3, and STAT3 induce in the muscle the production of, again, IL-6 to have active core of pathway, but at the same time is able to induce noggin, and not only noggin, there are other inhibitors of BMP that are induced that blocks the action of BMP. At the same time, the cancer can per se, we don't say that noggin is only a muscle cytokines, but it can be also released by secreted by cancer. Noggin blocks BMP, BMP pathway. BMP now that are blocked cannot signal anymore to SMAD158. Now there's no anymore inhibition on the atrophy program that start to be active in muscle-inducing protein breakdown, the removal of sarcomeric protein. At the same time, BMPs have a trophic action also on neuromuscular junction. And you start to have a denervation phenotype and lose of the contact site between the predator synaptic that leads not only exacerbation of atrophy using atrophy, but also a condition of fatigue due to inability to stimulate properly the depolarization of the plasma membrane. Okay, so we can say that at least for weakness and fatigue there is also an important contribution to muscular junction. But it's not the only one. Now I want to a little bit underline the other aspect that is pretty important that I want to touch, mitochondria and metabolites. And this is because mitochondria are crucial in the cell, in different cells, not only muscle. But in muscle, they have to generate energy, and muscle cell is a post-mitotic cell, cannot divide and dilute the damaged mitochondria. And the network is very complicated. So it's a 3D reconstruction of how is the mitochondrial network in a muscle. You can appreciate the subcirculemma mitochondria, the perinuclear mitochondria, but also the intermicroglial mitochondria. And you can really see how is interconnected the network. It's a fantastic work of imaging. And Bonetto and Teresa Zimmers published in 2016 the first, at least, the proteomic analysis of which are the proteins that are downregulated or upregulated during cancer cachexia. And one of the most important hits is that most of the protein are mitochondria, respiratory chains, TCA cycle, glycolysis, are downregulated. The only few that are upregulated are the red ones. So there's a general suppression or alteration in the food pathways that are important to generate the food for mitochondria and mitochondrial protein, especially of the respiratory chain. And among the other parts that have been also looking at in that paper was also the proteins involved in mitochondrial dynamics. So protein fusion and fission events that mitochondrial dynamics together with autophagy are important for the mitochondrial quality control. Almost the two of the most important parts is important for maintaining a mitochondrial network in good quality and good performance. And not only in cancer, but also during chemotherapy, there is the downregulation, especially of OPA1 compared to other mitochondrial shaping proteins. So we were wondering whether mitochondria can be another piece of the puzzle that promotes atrophy and, especially in this case, fatigue. And I want to focus on mitochondrial fusion and fission, so shaping machinery. So mitochondria shaping machine is important for the mitochondrial dynamics. Mitochondrial dynamics control the fitness of mitochondria in order to optimize the function to produce more energy and less ROS, or a more physiological level of ROS. And more ATP using the possibility to form super complex assembly by changing the morphology of the crystal. On the other side, it's also important the shift, especially for the fission machinery, to fragment and remove the damaged part of mitochondrial network in allowing so the autophagy, named mitophagy, to remove the damaged area and maintain healthy the remaining network, part of the network of mitochondria. So fusion events are mainly due to two proteins on the outer membrane, mitofusin 1 and 2, and one of the mitochondrial inner membrane, OPA1, is also important for mitochondrial crystal shape. On the other fission machine is mainly triggered by recruitment of this GTPase, GLP-1, by different receptors, and so GLP-1 is the critical then player for the fragmentation of mitochondria. And I want to focus mainly on OPA1 because it is one of the more exercise dependent and one of the proteins more between a different fusion and fission that's downregulated in different conditions. And we generate inducible muscle specific OPA1 knockout animals by crossing OPA1 flux mice with a transgenic animal express the recombinase under a muscle specific promoter and fuse with estrogen receptor, modified estrogen receptor sequence. So only muscle CRE is expressed and the protein is stabilized when you treat the animals with an estrogen analog called tamoxifen. Now the CRE can go and recombinase the 2-loxP side, removing OPA1 in muscle, not in the tissue. And the experiments were pretty simple. So we leave animal growing till they become 5 months old animals, and then we delete acutely OPA1 in muscle specifically. And we look what's happened to the animal. So first of all, from the treatment, the animals after 1 month, they start to spontaneously losing body weight. At least they lose body weight because they lose fat, white adipose tissues, but also especially because they lose muscle mass. So after 3 months, this is the phenotype of the animal, is a cachetic phenotype. Muscle are almost disappear. This is a gastrocnemius, there is no muscle left because we dramatically decrease the fiber size, but also the fiber number. 50% decrease in fiber number, 50% decrease in fiber size. And among the different genes that we did when we did the transcriptomic analysis of the messenger pathway, again, noggin came up. So even this condition, when you change or alter the mitochondria, you have an upregulation of this by a real-time PCR with quantify that indeed the noggin was induced. It was induced from the early phase of muscle loss when the animals start to lose muscle loss to the late phase when they already are cachectic and they are really in a bad shape. Not only we have an upregulation of noggin, again, in the blood, in this case in the blood, we have an upregulation of all the inflammatory cytokines, including IL-6. And IL-6 was also induced as a transcript in the muscle, but not in the fight. And the animal, they die within 3 minutes. So OPA1 deletion recapitulate a condition in which there is an upregulation of inflammatory response, a muscle loss, dramatic muscle loss, like I said, with the animal death. So the question is, which is the link, the mechanistic link between OPA and lifespan in this case, animal death. And the top one gene in transcriptomic analysis was this cytokine FGF21, is 1,400-fold induced compared to the wild type. And it was not, it was time dependent, so we reached 15,000-fold induction at the end of the life of this animal when they are really cachectic. But not only a muscle, this, so there was also a 4,000-fold increase in the blood. So there was dramatically increase, not only as a transcript, but as a protein in the blood. So that was pretty convincing data that probably FGF21 should play a role in the phenotype of this animal. And to address this point, we generate a double muscle-specific inducible knockout in which OPA1 and FGF21 was simultaneously deleted the only muscle. And we look at the phenotype. Surprisingly, at least for me, there was a minor sparing in the loss of the body weight of these double knockout animals, mainly because you spare white adipose tissue, but the phenotype of the animal was much, much better. And the inflammatory cytokines, there were no more systemic inflammatory response to the blood, and the animal do not die anymore. So these animals are lean, but they are healthy, and they do not die anymore. So which is the link between mitochondria dysfunction of FGF21 and the systemic inflammatory response we do see in this animal? So keep in mind that, and come back to the autophagy, the mitophagy system. So keep in mind, as I mentioned, the mitochondrial dynamics of fusion and fission are important, and especially fission events are important for the fragmentation and removal of the damaged mitochondria. So in our cell, every 20 minutes, an autophagy is formed and is delivered to the lysosome, where it is degraded in few minutes, the content of the carbon. And this happens continuously in the cell. But recently, Romanello et al. In the lab showed that FGF21 is able to trigger an excessive of mitophagy. So if you have too many autophages that are formed in a certain small amount of time, you have an overload of the lysosomal system that now is not able anymore to degrade all the cargo. And for mitochondria, one is specifically important is mitochondrial DNA. If it's not degraded, now pieces, fragment of mitochondrial DNA can be released from the lysosome and can be censored by toll-like receptor number 9 that can activate an FKB pathway and can induce, for instance, IL-6 production that can contribute to an healthy aging, or in this case, cachexia. So if this is true, by reducing autophagus inflammation, by knocking down IL-6 is a marker, an important for autophagus inflammation, for membrane commitment autophagus inflammation. If we reduce autophagus inflammation, now we should not have any more lysosomal overload and exhaustion, and now we have no release of mitochondrial DNA, and all the pathways should be down. So that was the experiment. We knocked down LC3 in our OPA1 deficient animals, and we will check IL-6 expression as a transcript. And as I mentioned to you, in OPA1 deficient animal, IL-6 is induced, but by knocking down, we blunted this induction. So if the pathway is true, now we should be able to reduce IL-6 expression just by blocking toll-like receptor number 9. And so when we knock down again in our OPA1 toll-like receptor number 9, again, we reduce significantly the upregulation of IL-6. So if NF-kappa-B is the downstream target for important IL-6 expression, by blocking NF-kappa-B, now we should IL-6 block IL-6 upregulation. So when we overexpress, in this case, we overexpress IKB superrepressor, so it's a mutant which is not anymore phosphorylable, so it's not anymore degradable, phosphorylable by AKK. And you completely, in this case, block IL-6 upregulation in OPA1 depletion. So on this picture, it's pretty complicated. We have to add other players that are important. So in cancer growth, now mitochondria start to be compromised, probably because OPA1 and maybe autophagy is not controlled. The upregulation or dysfunctional mitochondria can produce ROS and can signal to the nucleus to induce again nodular. And so if you have another pathway, it can contribute to nodular upregulation in the interstitium of the muscle. At the same time, ROS can damage myotubular component and can contribute to fatigue as well as the less ATP production. And in this case, also to protein breakdown due to FOXO 134 overactivation due to ROS production in protein breakdown. On the other side, on the other pathway, mitochondrial dysfunction trigger ER stress that activate an unfolding protein response is sufficient that induce transcription factor family K94 that induce expression and secretion of FGF21. And FGF21 has a double action, lipolysis, contribute to the cachexia. At the same time, to the receptor can signal and induce a loop in which activate mitophagy, maybe compensate to remove the damaged mitochondria, but in this case, induce an excessive mitophagy, leading to overload exertion of lysosomal that leads to a release of DNA fragmentation, the fragment of mitochondrial DNA that activate a toll-like receptor and FKB pathway that can be also activated by ROS production. That signal to the nucleus to, again, another feed-forward loop to induce IL-6 expression that contribute to the signaling in the muscle beside the cancer and also contribute to a muscle lookup to IL-6 signal and start pathway. So this is just a small piece of the probably much more complicated puzzle that can be, because I had never mentioned satellite cell, never mentioned exosome or other kind of pathway that can contribute FAPs and to the muscle loss. But maybe for the fatigue and for the, that seems to be pretty important. And the message is that just so restoring muscle component and protein can be not sufficient maybe to reactivate a perfect force generation, because you need to take care about also reactivate or restore a normal muscular junction. You need to take care about also about mitochondria for the energy purpose. So I believe, I think that for the aspect that we have talk about not only atrophy but also weakness of fatigue. Now we have at least two possible candidates: alteration in a muscular junction with decrease in action potential, alteration in mitochondria that can generate less ATP ROS production, and mitochondria can feed forward, activate or alter, contribute to alteration in neuromuscular junction, and these altogether contribute to the dramatic phenotype that are present in cancer patients. I haven't touched this aspect, but for sure there's also a contribution about calcium and ions to the fatigue and the weakness in cancer patients. And this is the most important slide. I have really privileged to work with a current group of persons. All the story about muscular junction and the nodular population has been developed and performed by the postdoc Roberta Sartori and by Sandri. And the story about OPA1 inhibition and the effect on mitochondria on muscle atrophy has been performed by Caterina Tezzer, at the time PhD student, and the senior staff scientist. And I have to thank a lot of people. I have to thank a lot of people that helped and contributed to my research. I want to really thank you, the surgery, the Department of Surgery, in Stefano Mediano and Sperti, the people, the physiology and neurophysiology around Michigan for the analysis. Paul Gregorevich, which will share the virus and the data of C26 in this derivation. Fabio for the help for all the cancer cachexia. And thank you, thank you for your attention, and I'll take your questions.
So many thanks, Marco. It was really a great talk. I have to say that we had more than 110 people following. So the interest was really high and great, and the data, I think that really satisfied the audience. If I can start really just with a comment, and then I will leave the space to the questions. I noticed that you, being a guy that have focused for long on the control of muscle mass, now you're moving on the control of muscle function. And if I think at my research, I've been in cachexia for the last 20 years, and I have seen no drugs, no effective treatments coming on the market for treating cancer patients affected by cachexia. I want to ask you, and being a bit provocative, that maybe we were looking at the wrong target. Maybe we should start with the function, either metabolic function or the ability to contract and to do a workload. And maybe we will find that maybe in the early future, we will be able to provide a treatment for cancer cachexia. In the meantime, I asked the audience if you want to propose a question to use the chat, and then I will repropose the question to Marco. So please, Marco.
You are completely right, Fabio. But the beauty of the science is that you cannot predict what is the result of one experiment. So when I started to work in muscle wasting, I was sure that just, let's say, protein breakdown can contribute to muscle loss. And think about it, that I was also applying to a grant, say that blocking protein breakdown, autophagy protein, you do rescue or improve muscle loss in sarcopenia and aging. And in this big EU grant I got, all the experiments were just focused on blocking autophagy and UPS to prevent age muscle loss, At the end of the grant, we discovered the opposite. So that if you block the proteasome and the autophagy, actually you worsen aging and sarcopenia. So the beauty of the science is that it is really unpredictable. And I was thinking that it is not the correct way just to block protein breakdown, because all the clinical trials so far, for instance, with myostatin inhibitors, fail when you have to measure muscle function. Maybe they restore or they prevent muscle loss. They have even muscle growth in some conditions. But when you measure muscle function, they do not have any physiological effect. And so now we know much more about signaling pathways. And I'm looking at interaction between different organs in the control of the metabolism, in the control of the muscle and nerve interaction, in the control of muscle and bone interaction, in the control of between the matrix and the muscle fiber. Because we are usually, we have to simplify we try to simplify as much as possible in order to, let's say, be focused and have a story get published. But when I'm a physician, so I know the complexity of an organ and how an organ, not a cell, but an organ that is done by many cells, interacts with the other organs. So in my future, I would like to have a more comprehensive view in which all the pieces of the pathway are not only protein degradation, but is the connection between protein degradation and metabolites, muscle, nerve, muscle, vital tissue, muscle, liver, muscle, and different organs.
Yeah. So that is really instructive for the young. So just to know where they get a move. So the first question for you, Marco, comes from Dennis Gattrich. I don't know whether Dennis want to propose, or in the meantime, I will try to read the question for you, Marco. Good to see you again, first. And then regarding your denervation data, do you see the mechanism of knocking expression and loss of innervation in cancer cachexia different from aging? You mentioned the aging, but the point is that in aging, the tough is that the effects are at the neuromuscular junction where the nerve pulls away to induce atrophy. On the contrary, in your model, you argue that knocking induces atrophy first, and then the nerve pulls away. Is it correct? Thanks.
Then you are mentioning a very important point. It's difficult to say if this is the mechanism is different. So when we measure the motor neurons in our C26 animal mice, there was no difference between the sham, the control, and the C26, suggesting that the innervation happens mainly locally at the synapse. So you lose the contact, but you do not lose the motor neurons. While during aging, we do know that the motor neurons are lost in the spinal cord. But to tell you the truth, is not so simplistic view, because autophagy, all the time which an animal model will alter autophagy, either autophagy deficient animals, either because we manipulate a mitochondrial pathway or other pathway, we do see the innervation phenotype. So it's possible that one common point is that in both conditions, maybe there's autophagy dysregulation, either too much or too less, in aging and in cancer. That can trigger an alteration in secretion of an alteration in neuromuscular junction. In aging, you have also a contribution of motor neural loss, but probably locally there can be a very similar path. In both the conditions, we have IL-6 upregulation. In both the conditions, we have mitochondrial dysfunction in cancer and in aging. So some commonalities there are. I would not exclude that some of the pathogenetic mechanisms are shared between aging and cachexia. Cachexia are more exacerbated faster than aging.
Okay, thanks, Marco. So I will skip all the congratulations because there are a lot in the chat. So all agree that your talk was fantastic. And now I propose you a question from Elia Angelino. He's asking again on the neuromuscular junction, and he is telling that the sympathetic nervous system is really close in contact with the NMJ. So do you have any idea on whether its activity is affected in cancer cachexia?
I haven't looked at the sympathetic neuron. I know another lab is looking at. So there is another lab working on sympathetic nervation. And I cannot tell you because they are confidential data, but there is an impact also on that data.
Okay, so if you can move to the second part of your talk, there's a question about the relevance of OPA deletion on tumor growth. Do you see any effect when you delete OPA and you inject the tumor?
So in this case, we're OPA knockout animals that we analyze. So we just found that OPA1 was decreased in the muscle of tumor-bearing mice. So in that case, there was no tumor. So in OPA1 knockout animals, there was no tumor. It was just a knockout.
Yeah, sure. Yeah, the point, maybe if I can extend the question, relates to the fact that sometimes it is difficult to distinguish between systemic effects happening on the host and tumor effects. And if I can even go further, I noticed in your data that many alterations, and especially in cancer patients, are already present, even if cachexia, according to the current consensus on the definition and on the diagnosis, is still not present. So we should be really careful when we consider systemic effect, when we treat with a drug that we are targeting the muscle, but also even the whole body metabolism, the tumor, and so on. So it's sometimes hard to distinguish.
But also in animal model, the alteration of muscular junction was preceding the cachexia, the muscle loss and the cachexia phenotype. So for us, it's a condition that also contributed to the less activity or less performance in cancer, and this can have an impact on mitochondrial function. So one of the aspects that I haven't mentioned to you is that OPA1 and all the mitochondrial fusion or autophagy are exercise dependent. So if you have a problem in a muscular junction, you feel weak and you feel not so strong, you decrease your activity. And all these genes are suppressed and mitochondria is suppressed. Sure. And this can be a way also in which you start to have less. PH1 is another important activity dependent gene. It's one of the most downregulated. If you just be inactive for a few days, your level of PH1 transcript muscle drops dramatically. We have experienced in bed rest experiments in humans. So one of the possibilities is that this alteration in the muscular junction promoted by cancer can also impact on mitochondria due to the fact that you start to have some alteration in your physical activity. You are less performing, you are decreasing your normal activity.
Yeah, that's why we should always couple physical activity to any kind of drug or supplement or nutritional approach. That's my opinion. No, I'm not a physician. So Melissa Pupa is asking whether FGF21 receptor is differentially expressed in oxidative versus glycolytic fibers in muscle.
That's a good point. So in any case, I can mention that all these dramatically derivative fiber, but even in our hormone deficient fiber are mainly glycolytic fibers. So all the beta acids fibers are preserved or have less atrophy than the glycolytic. So we haven't checked in the different muscles, but for sure in the glycolytic fiber, FGF receptor are expressed.
Okay, we go back to something that you already commented, Marco, but maybe something more can be added. So Richie Dan, that is a physician, is asking whether blocking autophagy in humans, in cachexia, is it ready for clinical trial, or does this need to be better defined in preclinical models first?
So the problem of blocking, there is no inhibitors that are safe for the autophagy because let's say chloroquine, baphyllomycin are all lysosomal inhibitors and have important side effects. Even colchicine, all these are drugs that are used in any case for different disease. But patients that use colchicine to block the autophagism as in delivery, or chloroquine to increase the pH in the lysine, have a side effects important. There are some drugs that are available for activate autophagy, like tetbectulin or some others that are working like a spermidine. But really inhibitors, besides genetic approach, that can be specific for mitophagy or for autophagy, they are not yet available to Americans to go to clinical trial.
Yeah. I agree with you, unless Richard is going ahead answering that hydroxychloroquine has shown that can change LC3 levels in pancreatic cancer. I think there are distinct point of view and distinct way of interpreting the. We should move to the flux of autophagy, to the specificity, and many things.
Richard is right. So he's using cancer to treat cancer patients to kill cancer cells because autophagy is a survival pathway that starts to be reactivated when you have a cancer that is growing very fast. So, for instance, they are combining autophagy inhibitors plus chemotherapy to increase the probability to induce to survive and to kill the cancer cell. But in muscle, hydroxychloroquine is a chloroquine myopathy due to the fact that you block autophagy and lysosomal, and you induce like a myopathy is very similar to a glycogenic storage disease.
I definitely agree, Marco. So Anna from Craft Science is asking about the combination of targeting in general. Would you combine, you just described a couple of approaches, and in the literature we can find many. Maybe if we can move forward, do you think there's a hierarchy between the in the pathogenesis? So would you combine things, or would you start from something that?
I would for sure combine all the therapy. So it's like when you want to do rehabilitation from a patient that has like a muscle loss. So you start to do a nutritional and a physical activity. You combine two aspects, one mechanical loading and one nutritional approach. It's the same for this complex disease like this cancer, cachexia. You need a combined approach to promote protein synthesis, better reduce protein degradation, or better reactivate some type of protein degradation maybe. At the same time, ameliorate neuromuscular junction function, ameliorate mitochondrial function. And we do not have touch the aspect of the calcium homeostasis. So for me, the future is a personalized medicine in which each patient has to be consider in terms of the different aspects, how much muscle loss, which are the signaling pathways involved, and a combination of more approaches has to be, depending on the status. You have still a good electromyography, good neuromuscular junction, you just have a protein stimulus formulation problem or energy balance, you touch that aspect. You have both, you have to touch all the aspects, or you have all the aspects that are compromised, you have to touch all the aspects. For me, it is a combined therapy.
For sure, before personalized medicine is the answer, and we can't have the magic pill for everyone. And on this line, Richard Skipworth is asking why in his samples is not seeing morphological changes in neuromuscular junction in human cancer patients.
Yes, the other point is I haven't mentioned about the cancer type. So we have also like 40, 50 biopsy from esophagus cancer patient. That is what Skipper was looking at mainly. And all the signaling parts are completely different from pancreatic to esophagus cancer people. Also, the cachexia for me is different between the two different aspects. So, for instance, all the cancer people that have esophagus, they do not have autogene 1 becomes correlation. They don't have changes in the genes that are involved. There are almost no changes compared to what we have seen in pancreatic cancer. So, the other aspect is that what I show you is mainly for pancreatic and colon cancer. And maybe these pathways can be different or can have different roles in other cancer types. Plus, we have better statistics in terms of the number of the cancer patients analyzed. Almost around 100 cancer patients. And some trend can be also become significant when you increase the number. When we reach only 20 people for each cancer type, we never reach any statistical significance due to the variability in humans. So numbers and the cancer type can be the other aspect that can be the difference between our data and Skipper data.
Yeah, definitely. There was also another question on this point about already published data we all know on JCI that are on this line, and the point is the same. So changing the subject, Kay McLeod is asking whether inhibiting mitophagy would be better than inhibiting autophagy in general to reduce the side effects, to be more specific in the skeletal muscle alterations. What is your opinion?
Yeah, potentially yes, especially if you have a patient maybe that have a high level of FGF21, then there should be, again, a personalized medicine. Maybe in that patient, reducing mitophagy can be a way to also ameliorate the situation. It will be a case specific. Keep in mind that if you block out mitophagy and you have a drug that is going to all your body, you block also mitophagy in other cells, and maybe this is bad because if epithelial cells block mitophagy, then you can have a pro-cancerogenic effect due to mitochondrial release loss. So be very careful when you are talking about inhibiting mitophagy in this condition. I would say inhibiting downstream targets, so NF-kappa B potentially is a good candidate to block in the people that have high FGF21 and high mitophagy blocks.
Yeah, even inhibiting mitophagy is not free from side effects. Maybe it can provide the proof of concept for the treatment. So about the mechanism, but maybe for patients is not the best option. So 1 hour is gone. I think we can close and thank Marco for his time and for the wonderful data that he showed us. And I remind the audience that we're gonna have the next talk in February, always the second week of the month, and Marilia Silander is going to give the talk. So thanks, Marco, again, and all of you for being here.
Bye.